Today we will be talking about protein purification. So this is a chapter 5 material lecture. I initially wanted to skip this chapter because I thought it was only applicable to laboratory workers. So for instance, if you're not working in a lab or don't plan on working in research, I didn't think it would be important to know. But this chapter will be covered on the exam, so I'm going over it with you. We'll try to keep it very simple and concise, try to make this video short, but for now, let's just get right into it. So before you even start calculating any equations or measurements, you have to know that in order to begin the process, you have to break down the cell, right? So the cell has, some proteins in it, right? So the cell is a square and the protein is the letter P. Well, we have to break down the cell in order to get to the proteins, because let's say we want to measure the enzymes within the protein. Well, we're going to have to break it down, and that process of breaking down the cell is called homogenization. And you can do that to the cell by doing a number of things, right? So for instance, you can load it onto a centrifuge, spin it around really, really quickly for a long time, and the cell breaks, but the protein and the organelles within it actually stay intact. So when you do that, you have the free, you have like a freed version of the protein. Okay, so now let's assume that you have your protein and you want to measure how many enzymes are in it. Okay, so you have some tools that allow you to do that. And let's say that you start with a test tube with 3.8 milliliters of sample, actually 3,800 milliliters. Now within that little test tube, you have about 22,800 milligrams of protein. Okay. But how many enzymes do you have? Let's say that the total activity is a measurement of how many enzymes are in the sample at one time. So let's say that there are 2,460 enzymes, and let's say that enzymes are going to be represented with the letter E, okay? So within a 3,800 milliliter test tube, there is about 22,800 milligrams of protein. But we are interested in measuring the amount of enzymes. We do that through some sort of experiment, and we find out that there are about 2,460 enzymes. Right. But you are more interested in kind of understanding the specific enzyme. So within those 2,460 enzymes, you are interested in one enzyme that does a specific action. Okay, let's call that enzyme X. Let's call that enzyme X. And we want to know how many enzyme X's are in this sample. Okay, well, that's when specific activity comes into play. So specific activity is the activity of a specific enzyme. Scientists are really creative with their titles, but essentially it's saying, okay, well, out of this sample of enzymes, about this much percentage of that sample is your specific enzyme. It's weird to say, but let me try to say it as clear as possible. So what I'm saying is that the specific activity is essentially the total activity divided by the total protein. So for instance, if you want to find the total, if you want to find the specific activity of this, measurement, you have 555 enzymes per what? Per 14.5 milligrams of protein. And if you do the math, it's about 38.3. Okay. Well, it's saying that of this activity right here, of the 555 enzymes within the 14.5 milligrams of protein, there's about 38.3 enzymes that are of interest to you. So if you're looking for enzyme X, it's saying that 38.3 enzymes are going to be enzyme X within a sample of 555 total enzymes that is within a sample of 14.5 milligrams of protein. It's very difficult to say. It's kind of like a mouthful of words, but hopefully that does make sense to you. Let's go into percent recovery. So percent recovery is basically the total activity. So notice that total activity is similar in both these calculations, okay? So activity divided by the initial total activity. Well, what does that mean? Let's try to calculate the percent recovery for this sample. So it's saying that the total recovery is 275 for this sample, okay? And then we're going to divide it by the initial total recovery, which is 2,460. If you do the math, it's about 11% recovery. Okay, so you're probably saying, well, Brian, whenever we're doing lab experiments, we usually want to have a good percent recovery. We want to have like an 80% recovery. That is true, but think of it in this way. Let's say that you are Googling something. You're looking at something on the internet, maybe a pizza recipe. I'm actually hungry right now, so I'm thinking about pizza. I know it's not good, but I'm thinking about it. So let's say that you want a specific pizza recipe. Well, instead of looking on the internet and typing pizza recipes, you can type in margarita pizza recipe or 3 meats pizza recipe. And instead of having 5 million search results, you get 5,000 search results. Well, your percent recovery decreased, right? Instead of having 10 million search results, you now have, let's say, 11 search results. Let's just put searches. So instead of having 10 million or 100 million search results, you only have 11 search results. Your percent recovery decreased, but the specific activity, the specific recipe, the specific search increased. Look at what happened. So it said, well, if we're on this subsection of data, well, 38.3 enzymes are going to be the one that you want out of a sample of 555 enzymes, right? But if we continue to refine our searches by doing more and more experiments and using better tools, we can say that, well, out of 275 total enzymes, 152.1 of those enzymes are the one that we want of enzyme X. Now, we have 152 enzyme X's within a sample of 1.8 milligrams of protein. So essentially that, hopefully this kind of cleared up anything. You now know how to calculate specific activity, which is just a total activity, the total amount of enzymes in the system divided by the total amount of protein. And now you know how to calculate percent recovery, which is just a total amount of enzymes, let's say 275, divided by the initial total amount of enzymes, which was 2,460, okay? So hopefully that does help. I want to briefly talk about centrifuges. Okay, so centrifuge. Now, if you haven't worked in a lab yet, a centrifuge is a tiny little cylinder. Well, it's actually a machine where you open up the lid and you see a bunch of cylinders. You put in your sample into the cylinder, you close the lid, you turn on the machine, and the machine actually spins the cylinders really, like super really fast, okay? Like if the samples were alive, they would vomit. So I like to think of kind of carnivals when I think of centrifuges. Now, back in like the 80s and 90s, the carnival used to have this really cute ride where they would put you into the ride and you would strap yourself to the wall of the ride. It was kind of like this big cylinder, okay? And I can't really draw, okay? Because I'm an English major, actually English minor, but whatever. And we do not draw at all. So, anyway, so you would have like this little door, you would go in there, you would strap yourself to the wall. So let's say that there's a person right here. And this machine would actually rotate really fast. And now the centrifugal force would go here, let's call that C sub F, would push outwards, okay? And then your body would just kind of like fling to the wall, and it would go really fast, you would get sick. It was great. It's the same idea for protein analysis. So this is actually part of the homogenization. And the percent recovery is going to be like really high. So the percent recovery is like usually 100%. And you're probably thinking, wow, that's really good. Not really, because it's extracting all the proteins, which is a good thing, but at the same time it's extracting all the enzymes, which is not good because you want a specific enzyme. For this video, let's say that we are studying enzyme X, okay? Well, whenever you start doing protein quantification, you're going to always do centrifuge. You're going to put it through a centrifuge. Why? Well, the centrifuge actually allows cells to break and extract organelles. Okay, and these organelles actually have enzymes, right? Because organelles are like little factories. Factories have the workers, which are enzymes, which take some substrates and make products. So the enzymes theoretically live in the organelles. Now, if we can break the cell and release the organelles without harming them, you actually have little packets of enzymes, which is good. So the centrifuge breaks down the cell and releases all the organelles into the test tube. And then you could get the test tube and separate it even more through the methods that I will show to you in this video. We will now be talking about salting out. So salting out is a really simple test or method to do extractions. Okay, so usually done after centrifuge. Let's say that we have some test tubes, right? Say that we have some test tubes. And we're going to have some protein. So usually whenever researchers are doing this, their protein, the broken up protein is in like a liquid form. So let's say that this right here is going to be our protein, okay? So we have this, blah, blah, blah. This is our protein. Ooh, that is not a good color. And what a scientist is going to do is that they are going to add literally salt. So you can do ammonium sulfide or sulfate, or you can do like table salt. For this example, let's just stick with like the simple stuff. Let's say that we use sodium chloride, okay? And we're going to add sodium chloride to the test tube. It's a really wide test tube. And the idea in this experiment is that whenever you add the sodium chloride to it, the sodium chloride is going to pull some of the water from the liquid. So the protein has, it's in a liquid form. The liquid itself has some water content, and the sodium chloride is going to make some interactions with the water because it's polar, and is going to kind of pull the water away from the protein and leave most of the protein intact. So let's say that we have a mixture of something like this, right? Okay. And then we are going to have our protein. So as you can see, this is a pretty cool experiment to do, right? If you want to try this at home, you can get a bottle of vodka. Let's say it's 40% vodka, and you can actually put into a container and put a lot of table salt in it. Now make sure that this table salt doesn't have any iodine, okay? And what's going to happen is that the alcohol, the ethanol that you want to drink, is trapped within a solution of water. That's what it means to be 40% alcohol. out of 100 milliliters of water, 40 milliliters are going to be alcohol. Well, whenever you add salt to it, the salt is going to pull the water to the bottom and leave the ethanol to the top. And you just kind of separate the ethanol out, and you have more ethanol. Yes, the percent recovery is going to decrease, but the amount of alcohol, the specific activity, is going to increase. So instead of getting 40% alcohol, you may have 80% alcohol. Instead of having a liter of alcohol, you may have 500 or actually 400 milliliters of alcohol. Okay, so that's how you get a stronger proof for the cheap. Okay, so yeah, just make sure that you don't use iodized salt. You want to use just normal table salt to increase your alcohol content. Okay, so food for thought. So now we will be talking about column chromatography. Okay, so this is column chromatography. So chromatography. And column chromatography, we're just going to call it CC, is going to have two phases. Okay, so you have the stationary phase, and then we also have the mobile phase. Okay, so what is the difference between the stationary phase and the mobile phase? Well, the stationary phase is whenever you have a tube, let's ignore the sample, let's say that it's not there yet, filled with buffer. Okay, so you can see that the column is packed with a essentially a buffer, right? They say stationary phase, that's a buffer. It's kind of like the little liquid that you want to mix things in. Okay, so now we have a buffer in the test tube, and we start adding our samples. So let's ignore the first picture. Let's say that we start loading our samples in. Okay, so this was sample 1, this was sample 2, and this is sample 3. Now samples are going to be proteins. Okay, notice that nothing is moving yet. We're only focusing on this top portion. Nothing is moving yet. The proteins are just chilling, they're just relaxing within the buffer. That is called the stationary phase. Ooh, cannot spell. Stationary phase. So it's just whenever you load the column with buffer and then put your proteins in it. They're not doing anything, they're not moving, they're not interacting. It's very bland. Now, whenever you're done packing it in and you want to actually separate your proteins, you add more buffer. So let's put our buffers over here. So we have buffers right here. So we have buffer. Let's add it. Now, the weight of the buffer is going to push downwards. And when it starts pushing downwards, gravity is going to take over and continue to push down. Now, the first things that come out into the test tube is just going to be buffer. So we keep putting buffers into the test tube. Let's say that we only want to do 0.001 milliliters per test tube. So every 0.001 milliliters, we switch to a different test tube until we hit our proteins. Okay, so this protein right here was the first one that we loaded. It just happened to be that this protein had like the lowest molecular weight. Whenever you're doing this, it doesn't matter if you put green first or blue first or whatever. It's the one that moves the quickest. Okay, the one that has like the most weight usually goes down the quickest. So there you go. So now the protein, the red one, eluted. That means to come down into the test tube, and then buffer, buffer, buffer. Eventually we have sample 2 making its way into the test tube. Then we have more test tubes with buffer until we get the green one, sample 3. Now let's say that you're doing a lab experiment that has been done a lot of times, and you know that sample 2 is going to have your enzyme. Well, you can ignore sample 1 and you can ignore sample 3. You could just focus on sample 2, right? Because let's say that all these proteins came from a single test tube, but then inside the column they separated into different protein groups, and then you separated those protein groups into test tubes. So the percent recovery is going to drastically decrease because now you're closing in on the certain group that has the specific enzyme, enzyme X. right? So percent recovery is going to go downwards. The specific activity is going to go up, okay? Cool. Now we will talk about a subsection of chromatography. Okay, so column chromatography can have different modifications to it to make it even more efficient. Okay, so you want to increase the specific activity as much as possible, and you want to decrease the percent recovery as much as possible. Okay, so this one is going to be called affinity. Affinity chromatography. Okay, so here we go. Actually, this is not affinity chromatography. This is actually size exclusion chromatography, which is cool. So this is size exclusion. Exclusion, so that's a U and an L. Chromatography. Chromatography. And essentially, you want to get the sample. Let's say that we're working with sample 2, okay? And sample 2 has proteins that are very similar in weight. So within sample 2, let's say that there are 2 groups of proteins. Okay, so let's call that protein 1 and protein 2. Now protein 1 has a size, let's call that Daltons. Now Daltons is a unit of measurement, okay? So it's 1 atomic unit. If you remember from Chem 1, an atomic unit is like the basis of how big an atom is, okay? So a Dalton is just an atomic unit. So let's say that P1 has an atomic unit of, I don't know, like 6,000, okay? And let's say that P2 has a Dalton of 9,000. Okay, so they're kind of close to each other, right? And we want to separate P1 from P2, okay? Because let's say that the enzyme EX resides in protein 2, okay? So what we're going to do is we're going to pack these gels, well, these columns with some beads, okay? Some beads. And whenever you're working with proteins, the beads are made from they're made from acrylamide. So these beads, which are kind of like little jellies, are made from polyacrylamide, and polyacrylamide stops proteins, okay? So it binds with certain proteins, it stops them from moving as fast. It's actually the basis for gel electrophoresis. And we're going to get into that later on in the video. But the lab has these beads made from polyacrylamide. And essentially, these beads, right? These beads, let's draw kind of like a donut shape, have this tiny hole, okay? And this hole, let's say, has a Dalton of 6,000 units, right? That means that P1, whenever it's being in the mobile phase of the column, is going to actually be trapped within this little hole, okay? And it's going to slow down the process of going into the test tube. So notice that P2 does not have a Dalton of 6,000. It has a Dalton of 9,000. Therefore, P2, let's say that this is P2, it's just going to ignore the bead and go into the test tube. So this was P2, and this is going to be P1, right? So P2 entered or eluded into the test tube first because it had a heavier weight. Okay, so the bigger the Dalton, the heavier the protein. Since the protein cannot fit inside the beads, it will skip around the beads and enter the test tube first. P1, however, enters the beads and it slows down the process of entering the test tube. You can imagine it as hitting traffic, okay? So the P1 tries to go into the test tube, but it hits traffic and is slowed down, okay? It is slowed down. P2 ignores the traffic, it has some sort of special privilege, and goes into the test tube first, okay? So now, instead of having sample 2 consisting of 2 groups of protein, you now have sample 2 with a higher amount of one protein group. now one protein group. So you can see that the percent recovery is just going to go in half. It's just a ridiculous amount of reduction right there. But the specific activity, let's call that specific activity, goes way, way up, okay? Because within protein 2, you will now have a higher chance of getting enzyme X, okay? So this is pretty cool. Notice that the beads have different Dalton limits, okay? So depending on the experiment that you're working with, you can have beads that go up to 60,000 Daltons, 50,000 Daltons, etc. In this example, I just said it was 6,000. In real life, there's like different packets that have different limits. Now this one is actually Affinity. So this is Affinity Chromatography. Let's just call chromatography C, okay? Now, what happens here is that, let's say that within protein 2, okay, we have little tiny organelles, okay? Organelles that we don't want to bring into the system. So within protein 2, there are little tiny organelles that we want to get rid of in order to increase the amount of enzymes present. Well, what we do is we pack the column again with some substances, okay? I'll give you an example of this later, but let's just say that the substance S wants to bind with the organelles. Now, whenever the protein starts to go down in the mobile phase, that's when I add the buffer into the test tube, and the weight of the buffer pushes it downwards. So what happens here is that as the material passes down, the substrates, well, actually the organelles in this example, connect to the substance S, okay? And the proteins, they don't really like substance S. They actually hate it. So they try to push themselves away from the substance, depending on the experiment. And all you're going to get is the protein, okay? So now we have protein 2, which is more purified. So this is protein 2 purified. So that is just pure protein, pure enzymes, no organelles at all. Amazing, okay? So notice that as we keep doing these methods, we get more and more specific. We're going to decrease the percent recovery. If I say something a lot of times, most likely it's going to be on the exam. Whenever you decrease the percent recovery, usually the specific activity goes up, okay? And the total activity goes down, right? Okay, so now we have the purified protein, and depending on if you want to get a really specific amount, you can do it again and again using different substances that bind to different proteins, okay? So that is pretty good. Now, an example of this in real-world application is with alcohol, okay? So whenever factories are making alcohol, like vodka, they usually want to make their machines or their diffusers, right, out of copper. Why is that? Well, whenever you're fermentating the material, you have some sulfur in the material, and you do not want to be drinking sulfur. You do not want that to have in your product. So the copper in the machines actually bind with the sulfur, okay? So let's load our test tube right here. This test tube is made from copper, and the alcohol is mixed with, well, it has sulfur in it, right? So here's the sulfur, whatever, and let's say that this is going to be our alcohol, okay? The sulfur is actually going to bind to the copper and go away from the alcohol. And whenever we have, diffusion, the alcohol comes out in liquid form and leaves the sulfur in with the copper. So that is called affinity chromatography. So it has real-world applications, and if you have a couple of drinks, you can actually thank diffusion chromatography from keeping you safe, okay? This is a ion column chromatography, okay? So this is actually ion exchange. And essentially, all you're doing is that you notice that it's very... sometimes you won't use this depending on the protein. Like if your protein is neutral, you won't use this. But depending on the proteins, if it's positive or negative, you're going to be using the ion exchange method. Okay, so you're packing your column with these molecules, okay? So with ions, let's say that in this example, we're working with positive ions, okay? So this is positive ions. This is an example of an anion exchange. You're probably saying, well, that doesn't make any sense. If we're working with positive ions, why are we calling it an anion? We should be calling it a cation. Well, it's in respect to, so references protein charge. So it's whenever we say anion exchange, we're saying that the charge on the protein is negative. So we can say that the protein is an anion, okay? And therefore, the buffer is positive. So B is equal to positive, right? And likewise, whenever we say cation exchange, what does that mean? It means that the protein is positive, okay? So this right here is a positive protein. And that means that the buffer is going to be negative. So in that case, we would be using negative ions to attract positive proteins, right? So that's all you're doing. So yeah, the cation exchange, the negative buffer picks up the positive proteins and it puts them into a test tube. And in the anion exchange, a positive buffer, which in this case would be sodium, picks up the proteins and carries them to the buffer. This separates some proteins. So for instance, if you have a mixture of proteins, let's say P1 and P2, P1 is positive and P2 is negative, but you want to have P2, then you would be using a positive ion, okay? So the positive ion, sodium, is going to pick up protein 2, which is what you want, and put it into the test tube. Now, it's going to leave protein 1 into the test tube since it has a similar charge. This is gel electrophoresis, and if you've taken genetics, you've probably worked with gel electrophoresis. But the difference between biochemistry and genetics in respect to gel electrophoresis is that in genetics you were using an agarose. So you're using an agarose gel, and that actually binds to nucleotides, which is not the focus of this class. Instead, we're going to be using the acrylamide, which binds to proteins. What makes this really good is that polyacrylamide actually has a higher resistance for heavier molecules, okay? So more resistance for heavy molecules. All right, what does that mean? Well, whenever you read a gel electrophoresis, you read it from top to bottom. So top to bottom, okay? So we have some bottom right there. And it's saying that right here, when I look at this, I'm saying, well, over here, we know that this is going to be a heavier molecule. So this is heavy. And this one is going to be lighter. So this is light, meaning that it's not as big or bulky, okay? So let's say that this one has a Dalton of, let's say, 100,000. And this one has a Dalton of about 10,000, okay? So you can see that within the polyacrylamide gel, the heavier proteins are actually further up, meaning they didn't move too far, okay? But the smaller proteins moved quickly downwards, okay? So that's pretty cool. But what if, let's say, what if that we have two proteins that are 10,000 Daltons each, okay? How can we separate them? Well, you can separate them by their charges. So, for instance, you can have a negative charge right here and a positive charge. The negative protein is going to go further downwards to the positive end, which is here. And the neutral protein is not going to move at all, right? So we can tell the difference between charges, or we can actually kind of separate them by pH. So remember, well, let's say that this is going to be a 2D electrophoresis, gel electrophoresis. So 2D means 2 dimension. Okay, the first dimension is going to be focusing on the pH, right? So we have our proteins right here, and we have a column that has different pHs. So let's say that this is a pH of 4, this one's 5, this one has a pH of 7, this one has a pH of 10. Within these different layers, your proteins are going to have isoelectric points. So you remember in a couple of videos ago, I taught you about isoelectric points, and where I said, at different pHs, some proteins are neutral, some proteins are extra positive, some proteins are extra negative, right? Well, here we want to layer the pHs in order to get all the proteins at their isoelectric point. So, protein 1 might be neutral at a pH of 4. Protein 2 might be neutral at a pH of 5, protein 3, etc., etc. After you figure that out, you want to load it up into these wells. These are called wells. Okay, these little kind of dots right here. So protein 1 is going to be here, protein 2 is going to be here, protein 3, etc. Now this is assuming that they have the same size, okay? That they have the same size. So now we know for sure that protein 1 is there, protein 2 is there, and protein 3 is there, okay? And then we can actually kind of separate them out doing other methods. But the 2-dimensional gel electrophoresis is superior than just having to do the molecular weights, okay? Because it's an additional step that decreases protein recovery. Let's say that you're working on a project where you have to essentially get all your amino acids in the sample and try to put them back together, okay? So it's kind of weird to think about that, but that's kind of like the Human Genome Project, right? So they had all these data points from people, and they tried to rearrange the data points to make the genome, okay? So we're doing the same thing. We're getting our samples of proteins, and we're going to break them apart. So let's say that we have protein 2, okay? And we were isolating protein 2, and so now our test tube is filled with protein 2, okay? We want to say, okay, well, I don't know what this is made of, but I'm going to separate it into all its amino acids. So I know that protein 2 is made from alanine, isoleucine, methionine, different amino acids. That's after I break all the amino acids apart, okay? So that is step 1. So that's step 1, is to get your polypeptide chain and completely annihilate it. Just break all the bonds and separate the amino acids. Next, this is assuming we have enough polypeptide chains, by the way, okay? So now we get a fresh chain, okay? And we're gonna say, well, where is the N-terminus and where is the C-terminus? Okay, well, right here is the N-terminus in the peptide chain, and over here is the C-terminus peptide chain. I don't know if tyrosine is the N-terminus. I don't know if alanine is the C-terminus. All I know by looking at the polypeptide chain from a computer is that the C-terminus is right here. It ends on this protein or amino acid, and the N-terminus ends at this amino acid. And we know that this is, I'm not counting, this is like 26 amino acids, okay? I know that my polypeptide chain has 26 amino acids or whatever. The N-terminus ends at this specific amino acid, and the C-terminus ends at this specific amino acid. Cool. After that, we start implementing some cutting, okay, some cleavage. And essentially, we're going to talk about cleavaging the chains in the next slide. But right now, what I'm going to do is, okay, well, I'm going to have a specific molecule, okay, that is going to cut down this portion, is going to cut down this portion, is going to cut this portion and this portion, etc., okay, down the chain. And whenever I have those portions, I can say, well, I can run that through a computer and find out, oh, this is alanine. This one right here is arginine. This one right here is whatever. And then, if you want to get more specific, we're going to do it again. But we're not going to use the same molecule to cut. We're going to use a different molecule to cut it now, because certain molecules cut different things, right? So now this guy that we added in step 4 is going to cut here, is going to cut there, is going to cut here, and there, and there. And essentially, you get more data, and between step 2 and 3, between step 3 and 4, you're going to get all these data points and saying, well, this was cut, this was not cut, and then piece them back together like a puzzle piece. Personally, it's not my style. I would not like to do that, but some people like that. Okay, so we're going to be talking about how to kind of which molecules we use to cut certain amino acids. One of the molecules that we use to cut amino acids from the polypeptide chain is called trypsin. So trypsin, it cuts kind of like the C terminal bond, I would say, C terminal bond for arginine and lysine. Okay, so you'll notice that arginine and lysine are actually basic groups, okay? So these are polar, but they're basic. And that's pretty cool. Why is that? Here is a different picture of the same thing happening, okay? So whenever you have this chain, let's say that we don't know, let's say that we know that this is the N terminus and that this is the C terminus, and we're looking at it on a computer screen, okay? So you just see little tiny spheres linked together, but you don't know what type of amino acids they are. You just know that it begins here, it ends here. What is the amino acid? Okay, well, we can use trypsin, and it's going to essentially read, I like to say read, the polypeptide chain, and it says asparagine, alanine, arginine, serine, lysine, right here, and it says, wait, arginine. Well, arginine, I can actually cut that. I can cut that because I react with the R chains. Okay, so you can say reacts with R chains. R chains, or R groups, right? So it says asparagine, alanine, arginine. Okay, well, I'm going to cut that. And right here, we have the N terminus. Serine, lysine. Oh, I can cut lysine bonds as well. So I'm going to cut that too. So now I have a chunk that says serine and lysine. Now I go tryptophan, lysine. I could cut that as well. Serine, glutamine, asparagine, right? Leucine, isoleucine, arginine. Oh, arginine, I can cut that. And now I have a chunk right here. Threonine, tyrosine, and then the C terminus group. Okay? Now, whenever you have these chunks, you can read them individually on a computer, right? You can analyze it and it says, okay, well, instead of having sphere and sphere, it turns out that this sphere is a serine and this guy right here is a lysine. Okay, cool. So you can see kind of like the how valuable having these molecules are whenever you're trying to kind of like make the polypeptide chain, right? So whenever you're trying to describe it on paper, you can actually thank trypsin for cutting this big long chain into small analyzable chunks, okay? Another molecule that you can use is called chymotrypsin. So we have chymotrypsin. And what it does is that it reacts with aromatic compounds. Well, I guess you could say amino acids, right? And it's the same idea that it cuts at the C terminal group. So you could say it cuts at C terminal group. So what I mean by aromatic, I mean that it reacts to compounds that have this guy right here. Okay. Or rings, I would say. You know, if you want to even simplify even more, you have rings. So this includes things like phenyl. So phenylalanine also has a ring structure in it. Tryptophan for sure has aromaticity, so it has a cyclohexene. It also has the little thingy right here, right? So kind of looks like this. So that's also aromatic. I'm not really going to get into it too much, but just know that it reacts with tyrosine, tryptophan, and phenylalanine. For sure, like it's always going to react with that. So here's the N-terminal group. We don't know what these amino acids are, but we do know that whenever, let's say that before we did this, we tried to put in tryptophan. Okay, so let's see, tryptophan, nothing is going to react, right? Because tryptophan only reacts with arginine and lysine. Do we have arginine here? No. Excuse me, trypsin. Trypsin. So we put trypsin in here, and it didn't react with anything. It didn't cut anything. So we say, okay, well, let's try chymotrypsin. And chymotrypsin sees methionine, no. Tyrosine, oh yeah, I can cut that. So here we have my little piece. Then we go to leucine, no. Tryptophan, yeah, we can cut that. So we have a chunk right here. And we have glutamine, phenylalanine, oh, I can cut that right there. So now we have another chunk, and then we have serine and whatever, which is left over. So I think that's pretty cool as well. Okay, so you use them together. You use trypsin and chymotrypsin whenever you're trying to build the amino acid sequence. Another molecule, and that's all we're going to cover for these guys after I say this, is cyanogen bromide. So we have cyanogen. We have cyanogen bromide. Okay, which is like this. Right, so it looks weird, but essentially it's cyanide with bromine. Okay, and all this does is that it reacts, so cuts methionine. So let's imagine that we didn't have any rings at all. Okay, so no tyrosines, no tryptophans, no phenylalanines. We didn't even have arginine or lysine. Wow. So what can we do? Well, you can do kind of like a last resort, and you can put in cyanogen bromide. And what it does, it says, well, look, in this amino acid sequence, you actually had methionine. Okay, and I'm going to cut it here. And let's say that there are different methionines in that sequence as well. So now you have chunks of methionine that you can analyze. Okay, so this actually reacts with the sulfur. Okay, so that's all you have. You have trypsin, chymotrypsin, and cyanogen bromide to do the polypeptide analysis. Okay, so that's it for this video. Hopefully you now have a better understanding of protein sequencing and protein quantification methods used within the lab. You now know on the exam, if they ask you, what is a anion chromatography method? Well, you're going to say, okay, well, that is when you pack the column with a positive buffer to get a negative protein out of the solution. Okay, so you know the specifics of the methods, how it's applied, how it can decrease the percent recovery yet increase the specific activity, and you can do all that now, hopefully. Right, so I hope that this video helped you out and that you do well on your exams. And remember that I will always love you. Okay? So have a great day and be safe, and thank you for spending your time with me. All right, have a great day.